Hello, this is micro lecture 2 of chapter 17, and now instead of just looking at what are the different types of tissues and how do they all work together, what's the intro, we're going to look at one particular part. We're going to look at muscle contraction. And I feel most students have difficulty with muscle contraction because you need to know the definitions, all those things we just talked about in micro lecture number 1. We're going to start using some of these terms in this lecture. So let's get started. The behavior of skeletal muscle fibers, they have to have 4 major phases of contraction and relaxation. First, excitation. What's the process in which a nerve action potential will lead to a muscle action potential? What's excitation contraction coupling? This is going to be the event that links the action potentials on the sarcolemma to the activation of the myofilaments, thereby preparing them to contract, which is our next step, contraction. And this is when the muscle fiber develops tension and then shorten. Relaxation is when the work is done, the muscle fiber has to relax and return to its resting length. So what is happening here? And at the plasma membrane, an action potential has to move, or it has to propagate. So when neurons and skeletal muscle fibers have electrically excitable membranes, what happens, the depolarization and the repolarization, and if you remember those terms, depolarization is when it gets positive, repolarization is when it gets negative again, and then it goes back to resting membrane potential. And if you see that little dip right there, that's going to be hyperpolarization, and that is because the potassium channels close a little bit too slow, so it becomes more negative until the sodium-potassium exchange pump can bring it back up to the resting membrane potential. And it's because of this little hyperpolarization that makes it possible for the action potential only to move down the membrane in one direction. Because if you think about this, because of that little hyperpolarization, it's not going to go back to the one that just sent it because it would take too much to get it back up to resting membrane potential. And if you don't remember any of those terms, there are a couple of videos on YouTube that you can watch, but it's good to kind of see what's happening in this process. And sometimes listening to it isn't going to work for all students, but seeing a video or seeing something, an animated video of it, or somebody talking about it over an animated video would be a better way to look at it. So they spread along the plasma membrane in only one direction. This is generated in less than 2 milliseconds, so it's really, really quick. Travels only in one direction because of that refractory period, and that refractory period is caused by the hyperpolarization due to those potassium channels, and this is what allows rapid communication. The neuromuscular junction. So now this is where the neuron and the synaptic knob is going to come in contact with the sarcolemma from the muscle cell. And here you can see the motor neuron, you can see the synaptic knobs, and then you can see the actual muscle fibers. So it's located where the motor neuron comes in contact with the muscle fiber, and one neuromuscular junction per fiber. But each motor neuron may then branch out into multiple muscle fibers like you can see here. And this kind of goes back to what we already talked about, is one motor neuron can have many, many, many muscle fibers, up to 1,000, or it can do a couple, like in your eyes and your hands and your face, where you just need a little fine-tuning control. And it does that by how many synaptic knobs it has and how many fibers it can actually control. So now let's look at what is actually happening at the neuromuscular junction. At this junction, you're going to have the synaptic knob, which is going to have synaptic vesicles, and because it's a motor neuron and it's going to affect skeletal muscle, its neurotransmitter is going to be acetylcholine, and the acetylcholine is in red in this diagram. Then the next step is this acetylcholine has to do something. So in the synaptic cleft is where this acetylcholine is going to want to go. And in the synaptic cleft, you have enzymes that break it down. Those enzymes are going to be acetylcholinesterase. And then you're going to have the receptor that's going to bind to acetylcholine. And there is that synaptic cleft again with the acetylcholinesterase just floating around. So now something is going to come to the neuromuscular junction, and that's going to be via an electrical impulse or the action potential from the neuron. So that comes into the synaptic knob. It causes calcium. It's not in your book, don't worry about it. But calcium comes in, opens up channels. It comes into the synaptic knob. It binds to the synaptic vesicles and triggers the release through via exocytosis for the release of acetylcholine. Then once acetylcholine is in the synaptic cleft, it's going to diffuse and it's going to bind to the receptors. The receptors, once it's bound, are going to change the potential and the sarcolemma, which is down here, and sodium is then going to enter into the sarcoplasm. And by sodium coming into this cell, it's going to make it more positive or depolarize it. Once it's depolarized, it's then going to send another action potential down the sarcolemma. Then acetylcholine is going to diffuse away, and then it's going to break down by acetylcholinesterase. The acetylcholine receptor membrane channels will close, and then the action potential is going to move down. The action potential generated at the motor end plate immediately spreads across the entire sarcolemma. It's a very brief event. Once acetylcholine was cleared from the receptors, no other stimulus will occur until another action potential occurs at that synaptic area. So the action potential, once it's moving down the sarcolemma, or the outside membrane, it's then going to come to these T-tubules we talked about, or transverse tubules. And if you remember back to the first chapter, you remember transverse just means going across. So this transverse tubule is the network that allows this signal to spread quickly across a bunch of muscle fibers. Then sarcoplasmic reticulum is going to release calcium, and it does this by this action potential coming down. It's going to change the membrane of the T-tubule, and you have that terminal cisternae, which is that thickened end of the sarcoplasmic reticulum. And you remember that sarcoplasmic reticulum is going to have calcium. So what happens is it moves down. It's traveling right next to this membrane. This membrane is attached to the sarcoplasmic reticulum. And what happens once that charge changes the permeability here, it allows calcium to then enter the muscle fibers. So let's review. At the neuromuscular junction, you have the synaptic knob, synaptic cleft, and then you have the sarcolemma, or the muscle. And then what drug that blocks acetylcholine release, how would that affect muscle contraction? So if you had some type of drug that paralyzed the muscle, what it's actually doing, it's blocking acetylcholine from binding on to those receptors. If you block acetylcholine from binding onto the receptors, you'll have flaccid paralysis. Also, you can have something that doesn't clear acetylcholine from the synaptic cleft, and if you do that, then you would always have a contraction. So think about it. What would happen if there were no acetylcholinesterase in the synaptic cleft? Acetylcholinesterase could not remove the acetylcholine, so there'd always be an action potential signaling the sarcoplasmic reticulum to release calcium. If it keeps releasing calcium, your muscles will always be contracted, and that will be when your muscles are tight and not going to be able to let go. Now, a muscle fiber contraction uses ATP in a cycle that repeats during the contraction. So now we're going to look at how does the thick and thin filaments slide past one another. You might remember this is called the sliding filament theory, the sliding filament model. All of those mean the same thing. We're going to take these thick filaments and have them slide closer. And if you're moving those closer here, you're shortening the muscle fiber, and this is how you contract a muscle fiber. So first we need to start at a resting sarcomere. A sarcomere is Z disc to Z disc. So the zigzagging, that's going to be from here to here. There's your resting sarcomere. The myosin heads are all energized and ready to go. The caulking of the head requires ATP. So as long as you have ATP available, and if your cells are living, you're going to have ATP available, they're going to be ready to go. All they need is calcium to be released. Another thing to kind of think of, the myosin head act as an ATPase. So ATP binds to it, changes it into ADP and phosphate, and then that'll be attached to the head, ready to go. So what's happening? You see the myosin head up here. It has the ADP and phosphate. It's ready to go. All it needs now is the sarcoplasmic reticulum to release calcium. Calcium gets released. It will then bind to the troponin molecule, and you can see the troponin right there. The calcium will go over the troponin. It'll bind to it, and then what happens here is the troponin will move the tropomyosin, which is that string, and then it will have the active sites on the actin molecules be exposed. Once they're exposed, it gets us to the next step. And that next step is when the myosin head binds to the actin active sites. Because there's thick filaments on top and on the bottom, there are many myosin heads ready to bind to the actin active sites as long as they're exposed. Then they're ready to pivot or to create a power stroke. And to do this, they release the ADP and phosphate and pull it or shorten. That's what the power stroke is. It goes from here and shortens it a short distance. Continuing with the contraction, once it's already done its power stroke, it needs to be released. And to do that, ATP binds to the myosin head, releases it from the active site. Because they have the enzymes here, the ATP is changed into ADP and phosphate, and that recocks the head. And as long as calcium is present, it will rebind to the actin active site and continue the contraction of the muscle. Once it relaxes, the acetylcholine gets released from the receptor, and acetylcholinesterase will break it down to be reabsorbed into the synaptic knob. That means that once that signal is stopped, the calcium gets reabsorbed into the sarcoplasmic reticulum. Then it will then be stored again in the sarcoplasmic reticulum, and ATP is needed for muscle contraction as well, because what you need here, once the calcium is removed from the troponin, the tropomyosin slides back onto the actin active sites. You need the ATP to remove it from the actin active site. And then it can recoil and be recocked and ready to activate again. One thing to think about is when the myosin head is on the actin site, that active site in myosin is creating a cross bridge. So when you're going over this process, again, you can do YouTube videos, you can do, there's plenty of things on Khan Academy. I have a couple videos for the lab that kind of explains this, but it's good to kind of look at this through different sources. When you're doing the discussion for this chapter, take a close look at this and try to make sure you fully understand in that discussion and you put it in your own words so it makes a lot of sense. Because just memorizing sarcolemma, sarcoplasmic reticulum, T-tubules, acetylcholine, power stroke, recocking, actin active site, and all of these things, it's very confusing. So try to make a story that you can explain to your friends and family, and this becomes much easier for you to comprehend. And finally, what happens if you don't have any ATP? So when you die, you might have heard me say, as long as you're living and the cells are living, you have ATP. But if you die, your cells are no longer making ATP. So if you don't have ATP, the myosin head cannot be removed or disassociate from the actin active site. And if they don't become removed from them, then it causes stiffening. That stiffening without removing that cross bridge is called rigor mortis. It begins about 3 to 4 hours after death, and it's because the sarcoplasmic reticulum will release the calcium. The calcium re-enters the tissue where the muscle fibers are. The calcium will then bind to the troponin, which moves the tropomyosin and activates the myosin-actin cross bridge. And because there's no ATP, there's no way to remove that myosin head from the actin, so the muscles cannot relax. Rigor mortis peaks at around 12 hours and then diminishes over the next 48 to 60 hours. And the reason why it starts to diminish is because you have other enzymes also being released at the same time that are going to start breaking down the actual proteins that are going to be found in muscle fibers. So eventually the myosin head becomes no more because the protein that makes it up starts to break down. And that concludes this micro lecture. Good luck, and please try to use outside sources for this, because even though this was explained in under 20 minutes, it's probably going to take you over an hour to fully understand what's going on. Good luck.